Hello friends, welcome to the channel. In this lecture, we will be discuss about the colorimeter and their principle. The semi-auto and fully auto biochemical analysis are fully functional on the basis of colorimeter, which is based on the Beers and Lamberts law. Colorimeter it is an instrument that used for the measurement of color substance in the solution. It's an analytical system that calculates the absorption of specific wavelength of light. It works on principle of Beers and Lamberts law. Basically, colorimeter used for the measuring the color substance in particular solution or measuring the how much intensity of the wavelength of light in a particular solution. As an example, for measurement of the glucose in a patient sample, we take 1 ml GOD POD reagent or glucose reagent and 10 microliter the patient sample. By the chemical reaction, it produces the color or pink color. That pink color we measure. The intensity of that color can be read by the colorimeter, by which we can calculate the test result. The uses are to determine the concentration of the solution. Second, determine the reaction level. Biochemical laboratory for performing the biochemical test such as urine, serum, plasma, CSF samples. Then determination of bacterial growth, that means how much turbidity it seen, for which it can measure the bacterial growth of the culture. Test water purity, textile and food industry. Let's see the parts of colorimeter. At first, the source of light is tungsten lamp, then the slit, again the condenser lens, then filter, then sample cuvette or tube, then the light absorbance read by a photometer, which is further connected to the galvanometer. Here is the picture diagram of colorimeter. Here is the sample holder at which we add the cuvette or tube. The source of light is tungsten lamp, by which polychromatic light with the visible range arise. Then after a slit it converts to monochromatic light. Then by the condenser lens it convert to by the condenser lens it pass through a filter. Then it pass through a sample holder at which we place the sample, by which the light passes. According to the density of the light, it absorbs and some passes outward to the photometer and the reading taken by the galvanometer. Let's talk about the principle of colorimeter. The principle of colorimeter is based on Beer's law and Lambert's law. At first, we talk about the Beer's law. When a parallel beam of monochromatic light passes through a solution, then the absorbance of the solution directly proportional to the substance in the solution or concentration of the substance in solution. The absorbance A directly proportional to C, that means concentration of the solution. If we eliminate the directly proportional, the proportionality constant is K. A is equal to Kc. It's the equation number 1. Then Lambert's law. When a parallel beam of monochromatic light passes through a solution, then the absorbance of the solution A is directly proportional to length of the light path. Here is the length of the light path from here to here. Here A is equal to K is the proportionality constant and L is the length of the light path. If we combine the equation 1 and equation 2, we get A is equal to KCL. That means A directly proportional to C directly proportional to L. From there we can get the calculation formula. That is concentration of test is equal to optical density of test divided by optical density of standard into concentration of standard. OD means optical density and C is concentration. When we take the blank, then the concentration of test is equal to optical density of test minus optical density of blank divided by optical density of standard minus optical density of blank into concentration of standard. It is the formula for calculating the concentration of the test. Let's see the parts of the colorimeter. First, the source of light. Source of light is tungsten lamp used in colorimeter which emits the visible range, that is 420 to 760 nanometer wavelength of light. Then a monochromatometer, that selected filters placed in the pathway of light and test solution which selects a specific wavelength of light and pass through it. Then slit, a narrow slit placed to improve the quality of light. Then cuvette made up of neutral glass which passes the same absorption capacity. Then photocell, the photocell coated with the selenium that emits the electron proportion to the light falling on it. Then galvanometer, it measures the output of photosensitive element, means the light absorbed by the photocell, and the photocell converts the light to electron and sent to the galvanometer which gives the reading from which we can measure the absorption of the density of the solution. It's all about the colorimeter. If this video is helpful for you, then like, share and subscribe. Thanks for watching.